下面到了今天早上的最后一个 speaker，他是 ZIYAD 的 Dabour， 他是以色列泽法特学院教授，德宝品牌的创始人。Dr. Dabour 我要讲一些关于皮肤护理与皮肤疾病的药用植物与新技术产品的替代疗法。Dr. Dabour 先生出生于医药世家，也是以色列军方的首席医药官。他致力于并将现代制药技术与以色列古老配方完美结合。让我们听听他对植物护肤的独到见解。OK， 掌声鼓励，欢迎。
Hi, thank you very much for your invitation. My name is ZIYAD Dabour. I'm from Israel, and I will take you back to the nature, to the kingdom of plants and herbs, where, in my opinion, herbs and plants could be a very good microbial modulators and can preserve the skin microbiome and keep the balance of it and maintain the skin beauty and also the skin health. What I'm going to talk with you is about the alternative therapy by medicinal plants, and I will focus on psoriasis and eczema. Dr. Dabour is a laboratory based on the north of Israel in Galilee, in the mountains, and we are in the fields of olives and medicinal herbs. And what we are doing is the combination between nature and new technology and new science. Our mission is to give women in our villages a place to work. So, as you see, we are looking for natural and effective products and combining the natural nature, technology, science, and tradition. Our new technology, it works at the inside level of the cells and takes anti-aging and skincare products to a high level for repairing and protecting skin and hair. And all of our products are from the Galilee. The Galilee is the mountains north of Israel. And we are want to bring the old Galilee tradition to the 21st century. Our uniqueness is that we are using the gift of natural, of Mother Nature, surrounding and environment of the Galilee birthplace to cultivate eco-friendly, chemical-free manufacture cycle and products to maintain growth and environmental sustainability. So we are looking for scientific, vegan, medical device, skincare products, hair care products that are effective. So what we are doing is the vegan ethnobotanics effective products. And I met about 100 of old women and men in our area and interview them about the uses of the medicinal plants for thousands of years. And I took this knowledge and do research on our laboratories in Technion and in Bar Ilan University and in other universities in the USA. And we formulate our products to a wide variety of high quality hydrating, nourishing, and age-defining the products. As you see, we have a lot of lines that you can see here. I will focus on skin health products and in a product that we call it ProZ92. Some of our products are in China and also CFDA approved. So for thousands of years, Galilee old men and women have been using plant extracts for health and beauty. As you see, there is many herbs that used to use for psoriasis treatment. We can see here the picture, many of these herbs that used to use for psoriasis. And in the other hand, we see the traditional use of extraction and how they can beat these things. So we, this old man used the plants, the nature, for his food, for his healthy, and for his cleaning. And here we see the plants and the chemistry of the plant that we can combine these three things together. So the unique opportunity of us is called ethnobotany, and as we know, most accounts of plants used derived from literature is collected years ago from tribes, living groups without a written language or traditional use that is part of the culture. Those who would find and record plant use were faced with a daunting task, and we have to get the trust of the informants, get reliable information, keep from offending the tribal members, and publish this information where it could be useful. So this project from the Druze family, in which this plant has been known for over 1,000 years, and that was not disclosed outside of their family until recently. What is the Druze tribe? Druze tribe is traditionally located in the Galilee, in the Golan, in north of Israel, Lebanon, and Syria, and descended from Abraham. They have an eclectic religious practice that includes aspects of Islam, Christianity, Judaism, Buddhism, and other beliefs, of which very little is known to the outside world. Some are more secular than others, which is why we now have access to the ethnobotany of the Asphodylus species, the plant that we used to treat psoriasis. I will show you a little video. It's a short video about Druze tradition, how they welcome the newborn. They welcome a newborn, new baby, with herbs and olive oil and olive leaves, and do a massage, and it's the way that they welcome these babies. They are picking the olive leaves, olives, and also the grandmother go to the mountains and pick some herbs and put it in olive oil, lemon, and peppermint, and use a massage for a new baby to maintain it healthy, her skin healthy and calm. So Asphodylus, what I want to talk about, I will mention the history, the current use of the treatment, development, and validation of the herb for this. So who used it first? We don't really know, but the Mu'addi family, it's one of the oldest families in the Druze in Israel. It traditionally used these herbs for 1,000 years, more than 1,000 years. The plant was collected, and the family head would treat anyone who had a skin ailment like eczema, psoriasis, without cost as a gift. And the more modern family, Mu'addi members saw a possibility after I met him. He saw a possibility to both help sufferers of psoriasis and a business venture. You can see this picture before and after 8 weeks of treatment. So when the father died at 1992, the father didn't take money, refused to allow the product to be sold, and also didn't share what plant he used. Only after he did, his son shared us this plant, and we got involved with this product because there was a need to validate the use of this plant. And our goal was US IND for clinical trials as FDA asked us. So the approach was not easy. FDA didn't have a precedent for approving a natural extract. Products like Senacot, known since 700 CE, were already in use, but it's a mixture of senosides that is partially processed and not crude plant. What we want to do here is a crude plant, crude extract, and it's very hard to go through the FDA. So the pre-IND meeting with the FDA regarding what they needed to see was most informative. It recognized the need of the FDA to address natural formulations, extracts, for testing in clinical settings. No guidelines were published, and what we proposed to do was what the FDA eventually adopted for such products. And similar standards were met, such as showing stability for the extract, but rather than using a single identified material, we have to do the HPLC fingerprint to validate the batch-to-batch consistency. So we do the validation, and our question was the plant harvest in January the same as that harvest in August and May? And the answer, yes, and you can see here a sample of fingerprint of the constituents of this extract. So we talk about Z92. 92 is the year that the father was died. And Z92 is from concept through R&D to production. So we have to do in vitro trials, clinical studies, chemical analysis, safety tests, stability tests, and production. And as we are talking about the plaque psoriasis, plaque psoriasis is chronic multisystem inflammatory disease and predominantly skin and joint involvement. The plaque psoriasis could be in all the body, okay? And here you can see the normal skin. Where is the cursor? Okay, you see the normal skin and the psoriasis skin, and we see that there is a proliferation of keratinocytes on the skin, and this is what gives the scaly skin and the plaques of psoriasis. So the treatment for psoriasis till now, there is no cure for psoriasis, but there are multiple effective treatment options. Topical therapy is still the standard of care for treatment of mild to moderate disease. And current therapies include corticosteroids, UV light phototherapy, and extreme cases of monoclonal antibodies and chemotherapy. And these therapies include many side effects, and some of them are expensive and also often require considerable time to resolve the plaques. You can see here the algorithm of chronic plaque psoriasis. It could be topical phototherapy, and without phototherapy, it could be chemotherapy or biologic treatments. As you see here is the PUVA. PUVA is psoralen and UVA, and psoralen is from Psoralia coryllifolia, and it's, I think it's in Chinese medicine, and there is a combination between the UVA and the component active ingredients from the herb. the mode of action of the extract of asphodylus. This is the plant. You can see the flowers of the plants. And what's unique for this plant is that the underground organs, the asphodylus bulb, they have the ability to survive fire. So what remains is not reduced to ash. And it's very unique for this plant. So we took it and do the extract. And as we know, psoriasis is an autoimmune condition arising from the epidermis, where the keratinocytes prematurely mature, leading to skin growth about 7 times faster than the normal skin. And this is a response to inflammatory cytokines like IL-10. And keratinocytes model then are a good representation for plaque psoriasis. And we can, if we can prove that our extract can inhibit the keratinocyte growth, this would be a very good indication for a good effect against psoriasis plaques. We want to inhibit the keratinocytes without affecting the fibroblasts, that it's good for our skin and health. As you see, the skin, we have the keratinocytes, okay? Here is the keratinocytes and the fibroblast, okay? And we want to show that this extract can inhibit the proliferation of keratinocyte without inhibiting the fibroblasts. As you see, first of all, we study the effect of on fibroblast, and we see that this figure shows the growth of normal dermal fibroblast, where the uptake of titrated thymidine indicates cell growth, and we see no statistical difference was noted between control, our control, and this is our compound. Okay, our compound in different dilution. It's 1 to 5,000 dilution, 1 to 10 to 20 or to 40, and so we prove that this extract couldn't inhibit the proliferation of normal fibroblasts. In the other side, keratinocytes, we have here our control, and as we see, our product MC is product inhibits the proliferation, and in other fraction that we used, we wash it with other solvent to show the solubility of the ingredients. We can see more inhibition of keratinocytes. So what we did is 28 data points of each treatment. We collected it, evaluated, and using statistical methods to compare all cell growth rates. And the data indicate that intact Z92 and crude extract significantly inhibited the growth of keratinocyte, but neither had any effect on fibroblast. So the compound inhibits the growth of keratinocyte, and that this activity is specific for this cell type, that it's the reason of the psoriasis. We have, after that, we have to identify the active ingredients with the extract. It's not enough to show that it's active. So we have to identify the active ingredients, and the extract can be fractionated. It can be divided into fractions, and use column chromatography, several fractions to see the compounds. So the chemical work you can see here, we use it with Dr. Thomas Hartman from Rogers University, and Professor David Kinder from Northern Ohio University. And here you can see the compounds. We think that these compounds are the main active ingredients against psoriasis and against keratinocytes proliferation. This extract have another compounds that have antibacterial activity, and as I mentioned before, the psoriasis also is an inflammatory disease, so it can be synergistic with active ingredients for psoriasis. So active components are organic soluble, meaning they can penetrate the skin for useful treatment for psoriasis plaques, include previously described compounds for psoriasis and new compounds that they didn't found before. Complex compounds like flavonoids are abundant in the Z92 and likely to also have activity. We expect a synergistic effect from the compounds present. After that, we are going to human testing. Human testing we did by these trials sponsored by Taro Pharma. And we did two trials in 2003 and 2004. And we see no adverse effect or unexpected drug-related adverse events in all of the patients. The clinical studies, first clinical study was done in New York at Taro location, and for 8 weeks and 1 or 2 times weekly. The second trial was done also in the US and in Canada. And as we see that 68% of the patient experience an improvement of 1 or more on the physician's global assessment. We call it PGA. PGA, it's the scale for the severity of psoriasis, and we see that the product can lower this PGA. So the efficacy results, we can see the first trial. Here you can see the presence of the totally cleared or 70% cleared or 40 to 60% cleared. Here you can see the photos before and after. You can see before and after 41 days for the foot, and in the body we can see before and after 54 days. Here are other pictures. Okay. Before and after 70 days. It could take about 40 to 180 days. Okay, this is 130 days. And also we did the safety tests. Okay, and we did toxicity studies and in Celsius Laboratory Group in New Jersey, and all studies are compliance with GLP requirements of the FDA CFR. After that, after 10 years of research with 2 clinical studies, we found evidence that ProZ92 is a natural relief powered by technology for flaky, itchy, scaly, and problematic skin caused by different skin disease. It's good for psoriasis, it's good for eczema, and we are now looking for other disease. In 2015, we started to produce under GMP ISO medical device standards and marketed it as ProZ92. Since then, we have a growing demand for this product. So what's the future? The future, to where has this gotten? And as you know, as you see, PROZ92 registers as medical device in Israel, in Europe, and it's produced under medical device standards. So why do we need more studies? We are looking for the Chinese market, and we are looking for medical trials or clinical trials for approving these claims also for the Chinese patients. So in Israel, PROZ92 is included in Clalit and Maccabi, the biggest health providers and insurance in Israel. It's about 85% of the population in Israel are covered under these two institutes. Thank you very much.
All right, questions? 问题？
首先非常谢谢您的演讲。我想问一下，就是在那个临床测试的时候，为什么是要测试它那个 Z92 对那个角质细胞的抑制作用？这个是为什么要测试这个结果？
So the question is why in the clinical study you are going to test the inhibition of Z92 on the keratin cells? Why you tested that?
Keratinocytes, it's the cells that proliferated faster, about 7 times than normal keratinocytes in this disease. So we have to see that this product can inhibit this over proliferation.
Well, thank you. That was fabulous work. Now, psoriasis is a lifetime disease. Most of the people have that for life. Now, when you apply Z92 product to a psoriasis patient, I see that most of people within few weeks or months, they totally recovered. But if you stop using your products, are they going to come back or are they going to stay for a while?
Not all of the patients are cleared totally, okay? I mentioned it's about 23% that cleared totally. And we have patients for more than 5 years that it's cleared, okay? And sometimes, and many times, we have also psoriasis go back after treatment, because psoriasis is autoimmune system, autoimmune disease, inflammatory disease, and it's very connected to our stress, okay? And so it's not so easy. I mentioned that there is no cure for psoriasis.
That's right, yes. Now, did you combine your product with, write down the bio, such as antibody you was patient? Did you do the together works, see if this will increase the efficacy of the antibody
inside,
product?
Inside?
Yes. Yeah.
We want to give an alternative product, okay, without using steroids. Steroids is not so good for bone and for healthy people, also for infants and children. It's very risky. So we are looking to treat without drugs, without chemotherapy. Maybe we can do a combination with other things that I saw here about accelerating the immune system of the skin by microbiomes or something else.
No, what I mean is combined with such as Stellera, Humira, those kind of.
No, Humira and Stellera is a biological
Yeah,
monoclonal antibodies, and maybe it can be used, but we prefer to use topical treatment before reaching this chemotherapy and or biological therapy. Also, the Humira and other antibodies have their side effects, and we don't know for long term what the effects would be at the future.
Okay, thank you.
Thank you, Professor, for your good presentation in the morning. And I have two questions to you. First question is about the quality control of Pro Z92. As you said that we have the fingerprint for qualitative quality control. Besides that, we know that flavonoids are responsible active compounds behind its plant. So do you have any quantitative quality control based on you identified biocompounds from flavonoids? This first question. The second question is about the mechanism. As you said that the mechanism behind of the Z92 is related to intervention of epidermal regeneration. And do you have any prediction about the effect, the side effect on the normal skin? As we know that the epidermal regeneration is a general mechanism for normal skin. And for mechanism, we know that flavonoids have antioxidant bioactivity and anti-inflammatory bioactivity. So do you have any prediction about the mechanism behind the Z92? Thank you.
Okay, thank you for your questions. It's very long questions. First, about the first question, our fingerprint, as you can see here, is the fingerprint that can show the validation of the differences between the herbs that collected at January, May, or August to show that all the ingredients, the main ingredients, are present without the season of the picking of the product. So I show you the fingerprint of the components, okay, of the main components of this extract. About the second question, antioxidant is a common claim, okay? Everything that we're talking about is antioxidants, okay? But here what we are talking about, it's the inhibition of keratinocytes proliferation. And it's unique for this herb. And that's the difference. I know that flavonoids and retinoids and others have antioxidant activity and other components have antibacterial activity. But the main activity for psoriasis is the inhibition of keratinocytes. Okay. Thank you very much.